Hello everyone, I'm Mr. Furlong, and I got a question for you. What did the cell say to his sister when she stepped on his foot? Mitosis! In the last video, we talked about why cells divide, and we said that the DNA was unable to keep up with the demand of information that's needed to keep that cell running, and that there wasn't enough area for nutrients to get into the cell and for waste to get out of the cell. Well, in today's video, we're going to take a look at how they divide. And this is all done through a process known as cell division. Before we get into the process of mitosis, we need to talk about what a chromosome is. A chromosome is simply a long strand of DNA that is wound tightly together. It's kind of like this skein of yarn. I mean, this is just one long strand. There we got one end and there's the other end, and it's all tightly packed together. And chromosomes are doing the same thing in our cells. They wind themselves up so they don't take up much space. Now, chromosomes are slightly different in prokaryotic versus eukaryotic cells. In prokaryotic cells, we know that the chromosomes, because there's no nucleus, are just found in the cytoplasm. And of course, there's only one chromosome in prokaryotic cells. In eukaryotic cells, they come in pairs. So normally what we see is not this, but this. I mean, this is that one amount of yarn, but it's in an unwound state. So we call this chromatin. Now, the reason why we see DNA like this is because it's easy access to genes. And of course, genes is just a segment of DNA that's making up that chromosome. So again, we only see DNA forming into chromosomes during cell division. Now, scientists recently have been able to use a special type of procedure where they can color code chromosomes. And so here we see these X-shaped structures. And what we're really looking at here in these X-shaped structures, this is really one chromosome right there. Before mitosis begins, makes a copy of itself, or actually more technically it replicates itself. And they are connected here at the centromere. So really what we're looking at is not just one chromosome, but here's one chromosome, let's just say it's chromosome number 1, and here is an exact replica of chromosome number 1. So really what we have here are two chromosomes. And the reason why we have to have two chromosomes is because one chromosome number 1 will go into one cell, another chromosome number 1 will go into another cell. But we don't normally see DNA in this chromosome form. Normally it's in a chromatin form. So scientists using this same procedure has shown these chromosomes in their actual chromatin shape. And so you can see inside of the nucleus of this particular cell, all those color-coded, looks like balls of yarn all over the place. Each one of those is a separate chromosome in its chromatin form. Now, when we talk about mitosis, we're really talking also about the cell cycle. The cell cycle is the life cycle of a cell. And there are 4 distinct stages to the cell cycle. They are the G1 phase, which the G stands for growth, or this is the first growth phase. The G1 is the stage of the cell's life in which it's doing what it's supposed to be doing. So if this is a liver cell, it's doing one of the 500 functions of a liver cell. If this is a kidney cell, it's helping to filter out the blood. If it's a hair cell, it's being hairy, I guess. Most of the lifespan of a cell is spent in the G1 phase. And except for a handful of cells, like for instance brain cells, they'll just spend their whole life in the G1 phase. But at some point, as these cells are beginning to grow, they're going to be starting to get a little too big, and they're going to have to go through cell division, at which point they'll go into the S phase. S stands for synthesis, and in particular, we're talking about DNA synthesis. So in this process, the DNA is going to replicate itself, and we'll talk about that in another video. Once the DNA has replicated itself, it's going to enter into the G2 phase, or the second growth phase. During this stage, other organelles are going to be making copies of themselves. In particular, the centrioles are going to be making copies of themselves. Finally, it's going to enter into mitosis, or the M phase. Now here's the interesting thing. In the lifespan of a cell, the G1, the S, and the G2 make up what is known as interphase. Interphase can last anywhere from several hours to years, depending on the type of cell. The cell will only be in the M phase for anywhere from 30 minutes to a couple of hours, depending on the cell. And it's during the M phase is where that cell is going to split from one cell into two cells. Finally, at the end of the M phase, that cell is going to split into two cells in what is called cytokinesis. We start off with one parent cell, we now end up with two brand new cells. So let's take a real quick look at how this happens. Mitosis is the process of cell division, and it has four distinct stages. Those are prophase, metaphase, anaphase, and telophase. Now, I don't want to get into all the specifics as to what's happening inside of the cell. That's not all that important for us. But what is important is that we can see how the cell is dividing and that we can also identify these different stages. So here's an actual cell. This is a time-lapse photography so that this cell, which might have taken about 2 hours to divide, we're going to do this in about 1 minute. Here we see the cell in prophase. We can see the chromosomes, these kind of bumpy looking things here. Those are the chromosomes. Now, at the very beginning of mitosis, of course, the chromosomes had formed. Here we can see the cell membrane of this particular cell, but there's no nucleus. The nucleus is gone. It has disappeared during this process. Chromosomes have formed. They are attaching themselves to spindle fibers. Those chromosomes are going to attach to those spindle fibers. And once they attach, notice what happens. They all start to line up right in the middle of the cell. This is what we call metaphase. Now those spindle fibers are going to pull on those chromosomes to pull the two sets apart during anaphase. Notice the cell is starting to pinch off in the middle. This is telophase. And when it physically separates into two cells, we have cytokinesis. So this is a very fascinating process. We're going to spend some more time on this in the classroom, and I will see you then.